Hey everybody, welcome back to Bio229 Microbiology. I'm Mary Annarella, your host, and we are moving on. Now I have uploaded a new PowerPoint to our Moodle class, and you will notice that there are several slides here that I am going to skip right over. I've already told you we're skipping over chapters 10 and 11, so this, forget it, forget this stuff. Well, these are things that you've seen before, and we actually talked about a lot of this, like the 3-domain system. We talked about this already in class, so I'm not feeling the need to mull over it again. We talked about endosymbiosis, so you're not going to be held responsible for, using genotype or phenotype to classify organisms. We are going to move on to eukaryotic microbes, and we are skimming through this too. You guys are gonna love this, I'm telling you. So eukaryotic microbes, we still study a little bit. These include algae, fungi, which includes yeast, and protozoa. A lot of protozoa, you probably have heard of at least a few of them. So we will discuss these organisms. So let's start with algae. This part is going to be very PowerPoint driven. Okay, so most of what you need to know about algae, circle this slide right here. They are all photosynthetic eukaryotes. Okay, there are both unicellular and multicellular algae. But they are all photosynthetic. They all have mitochondria, and they all have chloroplasts, and they all have nuclei. So that makes them eukaryotes. What's important about algae that you know are a couple of things. I stumble over my words so much, especially as we get started, because this is just still so weird for me to be lecturing with no one here except me and my dog and, the neighbor's kids playing outside my window here. Anyhow, getting back to what we're supposed to be talking about. The names of algae end in phyta. Like, for example, they tend to. Like, rhodophyta is a red type of algae. So they're going to end in phyta. They are interesting, and they are, I want to say, ecologically significant for a couple of reasons. A lot of them produce toxins. So, for example, the organism that causes red tide produces a toxin that gets into shellfish, and if humans eat the shellfish that has been exposed to red tide, you're going to get really sick. And the thing is about the red tide on the East Coast is that if you eat shellfish that was harvested during a red tide, you can get really, really sick. If you consume shellfish that was harvested on the West Coast that had been exposed to West Coast red tide, it could kill you. It is much more toxic. The toxin for the red tide out on the West Coast, Pacific Coast, tends to be have a higher toxicity. But not all algae are bad. These phytoplankton, see the phyto? Doesn't end in phyta this time. This is an exception. It begins with one. Phytoplankton exist on something like 80% of the world's ocean surfaces. And because they're photosynthetic, they're recycling a lot of oxygen. They're putting a lot of O2 back into the atmosphere. I think this was like the first week in class I mentioned something about phytoplankton. And one of the reasons that it's really important to keep the ocean from being polluted is that a lot of the pollution will acidify the waters and make it inhospitable for the phytoplankton. And if the phytoplankton die off, we are going to lose a major source of oxygen on our planet. Not good news for us. Let me delete the screenshot here. This is to just show you a phylogenetic tree and of how related algae are, and they're not very related. This is based on stuff that we actually saw, that we would have talked about in chapter 10, so I'm not going to hold you responsible for this, but I just wanted to show you that they are not very related to each other the way we categorize them now when we actually look at their ribosomal RNA sequences. So not terribly related. Oh, here's a pretty picture. I bet you'll recognize this one up at the top here. It looks like a giant hamster ball with a bunch of little hamster balls inside of it. Volvox, that is Volvox, and a lot of us had to take a look at Volvox in a Bio107 lab. So if you took Bio107 at HCC, you would have had to have looked at Volvox up here. It is an algae. Down here is a rhodophyta. It's kind of purpley down there. So just some pretty pictures for you to look at. Algae are not plants. They lack the complexity of a plant body. They don't envelop their embryo in a certain type of tissue. They are not plants, but they are photosynthetic. All right, another set of interesting eukaryotic microbes are protozoa. Protozoa are not photosynthetic. So I have here that they are non-photosynthetic. Circle this slide. Big important stuff. Maybe we'll go with red here. So they are non-photosynthetic. They're mostly, strike the word mostly here, they are unicellular. Dun, 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 dun. There we go. So they are unicellular, and there are different classifications of them. And we classify them by the type of reproduction. We classify them by motility. My bad there. I was reading ahead and the wrong word came out my mouth. It happens to me all the time. We classify them by their type of motility. So for example, some protozoa are motile by using a flagella. That would put them into one category. They will exhibit both sexual and asexual reproductions, but mostly their lifestyle comes in two forms. What we call the trophic form, and you're going to get to look at some pictures of these for a simulated lab that we have for you later in this semester, or a spore form. Okay, now a spore form, I have it written down here, it's a cyst, or it's a resting form. Think of it as sort of like a eukaryotic version of an endospore. Okay, but not the same thing as an endospore because we're talking eukaryotes, not prokaryotes. So the trophic form feeding, this means it's actively metabolizing. The spore form or the cyst, this resting form, think of them as in hibernation. Like an endospore. The problem with protozoa is a lot of these cyst or spore forms will appear in the environment and get into, for example, a town's water supply. And it turns out that a lot of protozoa cause a lot of human diseases that tend to cause dysentery, which is a real drag. Dysentery, think really bad diarrhea. Before we get into that, let's just take a look at how closely related they are, and they actually are fairly related. They're all highlighted in red sort of down here. They actually are, the way we categorize them, they are evolutionarily related to each other. All right, so they're categorized by the type of motility that they exhibit. So I have some phyla here, and I will tell you right now, for the lecture exam, you are not going to have to memorize these. But for the lab exercise, you're going to need to make sure this stuff is all in your notebook, and we'll give it to you in another way as well. So without further ado, let's see, maybe green will show up. And actually in lab, we don't call it Sarcomastigophora, we call it phylum Mastigophora. Actually, let me erase that. Let's go with yellow. Phylum Mastigophora. And these are the protozoa that move by flagella. Now there should be another bullet right here for Sarcodyna, which move by pseudopods. So think amoebas. You know, pseudopods. They look like false feet, like that. They will actually move by pseudopods as well as grab food. Ciliophora move by cilia. The name is in, the clue is in the name. Ciliophora moves by cilia. Ta-da. Sporozoa doesn't move at all, but there's some real nasties in there. And then Microspora, they have these little polar filaments. In lab, we're going to have a little assignment for you for protozoa in about 4 weeks, 3 or 4 weeks. I want to say after Thanksgiving. So you'll get a little more experience with this stuff. But I am not going to ask you about, I'll say Giardia. Is that, how does Giardia move and what subphylum is it in? I am not going to ask you that stuff. But you do need to know that they are classified according to motility, how they move. Protozoa are interesting to us because they cause a lot of human disorders. So for example, Giardia, right here, is known to infect a lot of municipal water supplies. And if a protozoa if Giardia does get into a water supply, people drink that water, you get a Giardia infection, it causes like really bad diarrhea, really bad stomach issues. So one of the things that municipalities will test their water supplies for, not only for coliform bacteria, but also for things like Giardia. Giardia is also very common in beaver pee. And Giardia is one of the reasons you never, ever, ever drink water from a pond where there's a beaver dam. I mean, I wouldn't drink water from a pond that I didn't know had a reliable, spring-fed source to it ever anyway. But a lot of people who, would do a lot of backpacking before we had some really good water purification systems that were easy to carry into the back country, would just drink water wherever, and you could end up with Giardia, and it really, really stinks. There are also some protozoa that cause some sexually transmitted diseases as well. Trichinosis that is caused by a protozoa. The organism that causes malaria that is carried by the tsetse fly, that is a protozoa as well. So they cause human infection. Okay, so let's say we have done algae, protozoa. Let's move on to a very fun guy. Fungi. Fungi are a really broad, diverse group of organisms. They range from unicellular yeast, which is over here on the left. I'm pointing to it, but can't you see me pointing to it? It's over here on the left, the pink stuff. Unicellular yeast to multicellular mushrooms, which you can see here. Okay, so fungi. The study of fungi is called mycology, and their names tend to end in mycetes. Let's take another screenshot of this first. So like Ascomycetes, Basidiomycetes, they are non-motile, so guess what? We don't categorize them, of course, according to motility. We actually categorize them, or let's see, we categorize by the type of sexual reproduction. They do. What type of spores? Type of spores. Fungi and molds are also fungi. Molds are fungi, yeast is fungi, and the type of spores they produce is how we classify or categorize different fungi. All right, even though molds do tend to grow where it's damp, they live on land. They are not aquatic. They do have this substance called chitin in their walls. So it's one thing that they actually do all have in common, even though it's a very diverse group. But I do want to point out that they have a really filamentous growth habit for these things that we call hyphae. And it's one of the reasons why you can have a very difficult time getting certain fungi and molds off of a surface, because they will attach as a community these very filamentous structures called hyphae, and they'll be producing spores all along them. And it can be very difficult to remove from a surface. So these individual threads are called hyphae right here. So these are the threads. And together, all the threads form a mycelium. So this whole thing is a mycelium. Right there. Delete that. Let me show you a picture. All right, so here on the right, give you a nice close-up of all the hyphae here that look very filamentous, like that, and then some spores all over the place. And I don't know, I think we actually don't have the fungus activity for you this year, but some of them, some fungi will actually look kind of like, almost like this little branch, or with a bunch of spores coming off the hyphae like this. It looks kind of, I don't know, they say it looks like a broomstick. It's kind of a funky looking broom to me. So something like that. But they're going to have, a lot of them will have hyphae, and most of them will form spores. All right. Fungi do tend to be very related to each other. They're clustered up here in this one area. All right, classifying fungi. I'll take a screenshot of this, but I don't want you to freak out about it either, because I'm not going to ask you to give me an example of something from Zygomycetes or Basidiomycetes, you know, Ascomycetes, yeasts, and Deutero. But you do need to know that they are classified based on the form of sexual reproduction that they display. All right, so why do we care about fungi? Well, there's a reason we teach people not to eat mushrooms that they just happen to find in the woods without, you know, a guide who absolutely knows what that is. Ingesting certain mushrooms and certain fungi can make you very sick, right? And it's going to be different from the way that, say, people get sick from Giardia, from a protozoa infection. So let's talk about how eukaryotic microbes, these three, algae, fungi, protozoa, can affect human health. So, algae and human disease. Algae can make people sick by producing toxins, and then we eat the toxin, right? But you don't get an algal infection. No, that's not what happens. So algae can affect human health because we might consume a toxin that they produce. Protozoa, they cause a direct infection. And I already talked about Giardia, malaria, toxoplasmosis, that's the other one I forgot about, and a couple of forms of vaginitis, such as trich. Toxoplasmosis, that is a protozoa that tends to be prevalent in cat feces, and that's why pregnant ladies are supposed to not go anywhere near the cat litter box. You stay away from that, because you can get a protozoa infection, and you can actually pass it on to your baby. So toxoplasmosis is serious stuff, and it is caused by a protozoa. Right, so protozoa make us sick by direct infection. Giardia this first one here on that second bullet, that's the one that can infect municipal water supplies. When I was growing up, there was a neighboring town, and I went to a regional high school. And all of the students, all of my friends from that one town that had Giardia in their municipalities, I mean, it was like 6 years before they were able to get that out of their water supply. And I would ask my friends, I'm like, what do you do when you shower? And they would be like, well, you have to brush the water away from your mouth. I'm like, oh my God, that's gross. You brush your teeth with that? They're like, no, we don't brush our teeth with it. We have to get bottled water for everything else. And it was like 6 years before they were able to get it out of the water supply. Crazy. All right, so fungi and human disease. Now this is different. Fungi can make us sick, can affect human health anyway, from the spores that they produce. And a lot of people tend to have allergies against molds and mold spores, right? We can get sick from fungi by consuming a fungus that has a toxin, like a poisonous mushroom. Or they can actually cause a direct infection, such as a yeast infection, jock itch. Vaginal yeast infection is caused, yeast is a fungi. So this is called a mycoses, and you would not use an antibiotic against that. You have to use what's called an antimycotic. Like for jock itch and athlete's foot. Isn't it fast-actin, tenactin? That's an old commercial that used to call it that. So let's think about this for a second. Why would it be more difficult to treat a disease? Oops, I've got too many words in here. Why might it be more difficult to treat, cross out the second, to treat diseases caused by eukaryotic microbes rather than a bacterial infection? You want to think about that for a second while I have a drink of water? Okay, have you thought about it? Well, think about what we use to treat a bacterial infection. We use antibiotics, and think about what antibiotics target. Antibiotics usually target, oops, I can't spell. Prokaryotic ribosomes and or peptidoglycan. Our ribosomes are ADS, and our cells don't make peptidoglycan, so if we take an antibiotic that messes those two things up, one or both of those two things up, they're not going to hurt our cells. It is more difficult, and I got to get this out of there, it's more difficult to treat an infection caused by a eukaryotic microbe because they're too similar to our own cells. I mean, for one thing, we can't use antibiotics against them, typical ones, because why would they care about something that messes up their peptidoglycan? They don't make peptidoglycan. Protozoas don't make peptidoglycan, so we have to explore other differences, which is a little harder. Definitely easier with bacteria. All right, and then of course, there is a third thing that we get infections of that are not caused by eukaryotic microbes, are not caused by prokaryotic microbes either. And those would be viruses. So we're going to be talking about viruses that infect bacteria and viruses that infect human cells. This is so different that we're working with just PowerPoint on this. It's like, I think I just have a better PowerPoint for this stuff right here. All right. But just as a reminder, and this is all old stuff for you all. You already have in your notes from an earlier chapter that viruses are non-living, obligate, intracellular parasites. And that word intracellular means they have to be inside somebody's cell. When bacteria infect a human cell, like if this is our cell here, we've got all this stuff in here, Bacteria can infect our cells. They don't get in usually. Usually they'll set up an infection on the outside. Viruses, on the other hand, need to get in you. Oops. A virus has to get in you. If this is a virus, this little green thing, it's got to get into your cells. Intra means inside. They require a host cell in order to make more of themselves. A virus all alone, by its lonesome, if that's its protein coat and its genome in there, if it's all like this, just completely by itself long enough on a surface, and yes, I know coronavirus has been found to remain infectious for up to like 20 hours, but let's say this sits around for, a year. It's not going to be infectious after a year. It's got to be able to get inside a host cell, or it won't be able to make more of itself, and it will be degraded. So just a reminder, nucleic acid surrounded, which can be DNA or RNA, surrounded by a protein coat, which is also referred to as the capsid. So you're going to see that word a whole lot more. All right, where do viruses come from? We do not know. Like, where did their genes come from? Well, maybe their genes came from, an escaped from some nucleic acid, from a dead, viral cell, and it somehow got packaged up into a protein coat. And the evidence for that is because, viruses are most closely related to the cells that they infect, and they're very specific to their host. There's also an idea that they may have derived from some intracellular prokaryotic cell that got ingested. So kind of like endosymbiosis. Maybe they just co-evolved. FYI, I am not going to ask you to spout these off for me on the exam. You do not need to worry about that. And this stuff is getting into the final exam material anyway. But I just did want to mention that we don't really know where viruses come from. They are most closely related to the cells that they infect, but we don't know. We don't know, or all of the above might happen. All of these different scenarios may happen for different viruses. So let's do a little vocabulary here. Viral structure and shape. I just want to mention that the term virion refers to a single viral particle. A single viral particle. So let's say, I've got my genome here and a protein coat, or the capsid. This is the capsid, right? Right there. This whole thing all by itself. I do not like how the erasing function works for screenshots, but whatever. But this whole thing is referred to as a virion. So just a little vocab right there. I'm going to be using those terms. All right, they can come in different shapes. So for example, you can have what's called an isometric shape, or a dome. And that's what I was drawing here. Here we go. This would be a dome, except, I'm drawing this flat on a piece of paper. I can't really draw a geodesic dome very well. Or it could be helical in shape, where this is the DNA or RNA genome, and the capsid forms a little helix around it. And then there's some funky, weird, complex shape, like the T4 bacteriophage that I talked about last time, where it looks like a dome, and then it's got this little helical part, and then these little spikes down there. So that's a complex shape because it's got a dome up here, it's got a little helical shape right here, and it's got, these are actually its protein spikes, which help it enter the cell. Protein spikes can occur on any other shape as well. Like up here on this isometric dome, there could be protein spikes here. So like coronavirus has the spike protein, and you'll see it, they'll draw it like that. and it's the spike protein that will bind to the ACE2 receptors on human cells. So these protein spikes here will help it bind to a cell. Help virus bind to a cell. So we've got that. Oh, just to look at these pretty pictures. All right, so here's a nice picture of a dome, isometric. So a little drawing here, and to the right of the drawing there's an actual electron micrograph. This is an adenovirus that's domed, and doesn't it kind of look like a little geodesic dome kind of thing? Or maybe, I don't know, a soccer ball with way more little hexagons there. Helical down here, this is actually tobacco mosaic virus, which kind of cracks me up because it kind of sort of looks like a joint. It looks like a tobacco cigarette. But nucleic acid that's inside the coat, and over here to the right of it is an electron micrograph of it. And then, finally, T4 bacteriophage. Really complex. Please remember that the term phage or phage, I don't know, are we being French here? Is it phage or phage? But phage or phage refers to a virus that infects bacteria. Phage or phage. Sometimes it's the psi symbol there. If I use the term phage, you know that we're talking about a virus that infects only bacteria. All right. Naked virus versus enveloped. Okay, I don't know what the heck happened there, but my iPad totally crashed. And I'll have to go back and look to make sure that it still did the screen capture for me. But let's just open things back up again. Here we go. It's working on it. Welcome back 1 minute ago. That's right. Damn right. Where the heck am I? Okay, coevolution. T4 bacteriophage. Naked viruses. It did not like me saying naked, I guess. Yeah, okay, I thought it was going to crash again when I did that screenshot. Okay, all bacteriophage are naked. What that means is that if this is a bacteriophage, right here, okay, yay, the last screen recording was saved to photos. That's good, and we are still recording again. All right, it means that if something is naked, it means it doesn't have an envelope around it. Animal viruses sometimes are naked, but sometimes have an envelope, or we say they are enveloped. That's where the word envelope came from. It means to envelop. So if this is a virus, here it's naked. It could be either a bacteriophage or it could be a naked animal virus. But if it has a lipid bilayer around it, and I'll just draw it like this. This is the crappiest looking lipid bilayer you ever saw. It looks just, I don't know, like curly hair or a old-fashioned phone cord. This would be the envelope. It's a lipid bilayer that surrounds the protein coat. So this in here is the capsid or protein coat. And out here, the fluffy curly looking thing is an envelope, and there is what's called a matrix protein inside of it that holds it all together. There can be more to it than this. There could be, for example, like on coronavirus. Coronavirus does have an envelope. It could have proteins on the outside of the envelope, like the spike protein, if we're talking coronavirus. So coronavirus is an example of an enveloped virus. It has an envelope, And it's also got these little spike proteins out there. All right, so let's just take a look at this quickly. Up here at the top, this is a naked virus. There is no envelope around it. The one below, B with the little green things, that has an envelope, and it has its matrix protein. The capsid and the nucleic acid are inside of the envelope. And that's just to show you that pretty picture there. Size. This isn't just a nice little graphic to give you an idea of the size of viruses. If this is a human red blood cell over here, I'm going to look how big this sucker is. Compared to that, E. coli is much smaller, but look how much smaller adenovirus or tobacco mosaic virus or T4 bacteriophage are. They're tiny. Coronavirus is around adenovirus size. It's around 90 nanometers. Really tiny compared to your cells, but the havoc that they wreak can be intense, as we all know. All right, so we're getting inside a virus, and a virus, again, at its most basic, is just going to have a capsid, protein coat, or and a genome, which is either DNA or RNA. Let's talk about what has to be in the genome. What genes does the genome have for viruses? All right. At its very basic, these are the three genes needed. These are the three genes that are needed at minimum in a viral genome. 3 genes needed in a viral genome. At minimum. A lot of them have more than these 3, but they got to have these 3 or they're not going to work as a virus. First up, they need a gene that has the information, the codons, to build its own protein coat. Otherwise, how is it going to get its capsid made? So it has a gene for its own capsid. It also has to have an enzyme to ensure replication of viral DNA. So it's going to be a polymerase here. And then it's got to have something to get it in and out of the host cell. Let's say in and or out. So if we're talking of viral genome here, and I'm not talking coronavirus's genome, oh my God, coronavirus's genome is complicated. I was learning up on it going, holy crap, I think my brain exploded. Especially because it does something really, really wild. But anywho, we're doing a basic one here. It's got to have something for its capsid. So I'll write the word cap. If this is its DNA, or RNA, really. Capsid. It will have a polymerase, like let's say, I'll put pol for polymerase, and then it's got to have something to get it in and out of the cell, and sometimes sometimes it's called GAG, the GAG gene. But we'll talk about that. Let's say LYS for lysing the cell, something to get it in and out of the cell. Coronavirus has way more than this, but this is the basic for what a virus needs to have in order to be infectious. Oh, here's this pretty picture of T4 bacteriophage. Isn't that cool? Especially this one down here. It's that complex looking one. I love this. I mean, come on, how wild is that? It looks like they're landing on E. coli. And what's really cool is, do you see this? Right here, and in here, and here. That's the viral DNA getting inside E. coli. Viral DNA. Actually, this stuff, the capsid, stays outside the cell in this case, but only the viral DNA gets inside for this T4 bacteriophage. Now remember, all viruses are different. So we'll be looking at how a few of them behave to give us an idea. So for example, some viruses take over a cell's machinery in order to make more of themselves, and they burst the cell. These are called lytic viruses. They lyse, which is another word to say burst, destroy the host cells in order to release new viral particles. That would be a lytic virus. Some viruses are not lytic. Some of them don't burst the cell. Others are temperate. That's the word I'm looking for. Other viruses only take over part of the cell's machinery and they don't burst the cell. So they will release, let's say, those viruses will get inside a cell, get themselves made, make a whole bunch of them, and then have the cell release those viral particles, release those virions without bursting the cell. Okay, we'll call this a temperate virus. Temperate. Doesn't destroy the host cell. Okay, but there are some caveats here. So let's take a look at this nice little graphic I have here for you. Wow, this is a very PowerPoint-driven lecture today. All right, let's stay to the left here. So we'll start up here. You get an infection, it gets inside the host cell, and you get disease. We're going to stay off of this right side for right now. All right, you can get more virus produced inside the host cell. Lytic viruses will lyse the cells. A latent virus will not lyse the cells, and it will just release virions. It will. But this part that's over here on the right side, let me just kind of erase this for right now. What happens with a lot of viruses is, and this is when they go latent, is that they will get inside a cell and their DNA will become incorporated into the host cell's genome by genetic recombination. So viral DNA gets incorporated into the cell's genome. That's when the virus goes latent. And I had shown you a little bit of that when we talked about transduction last time. But like, let's say there's a cell, and here, let's make it a bacterial cell, and let's say T4 bacteriophage gets its DNA in there. And the viral DNA by homologous recombination gets inserted into the cell's chromosome. This virus is latent. I know this is like, well, I don't see any viral particles there. The cell knows that's foreign DNA, actually. And this DNA here can get those genes expressed, and it can start turning this cell into a viral particle factory in a second. But while the cell is in this state with the little bit of viral DNA in it, the cell is called a lysogen. A lysogen. Things are latent. And while this little piece of DNA that is viral DNA, but it's incorporated into the bacterial cell's genome, it's not the whole virus, but it's got all the information to build the virus. So we say that this is like a pre-virus, or we call it a pro-virus, or in this case, a prophage, because remember, phage refers to viruses and in fact bacteria. So a provirus or prophage refers to a piece of viral DNA that has been incorporated into the host cell's genome. And you know what? Whoa, how did I get this? How did I get this ruler to come up there? That is new to me. I've never done that before. Let's write this down in clearer terms. Oh, eukaryotic microbes. We already did that. Let's talk about a provirus or a prophage. Okay, let's say we've got this bacterial cell here that has been infected by a virus. Let's say it's T4. And I'll give a little viral DNA a red color. Through the lytic cycle, T4 lands on E. coli's surface, cuts a little hole in E. coli's cell membrane, and the RNA gets inside. Sorry, the DNA. Only the DNA gets inside. I didn't want that. It erases the whole thing, so I'm going to have to go like this. There we go. Okay, so this is actually called uncoating because the capsid stays outside the cell. Only the DNA got in. And then through homologous recombination, the viral DNA can be incorporated into the host cell's genome. There we go. This little bit of viral DNA is called a provirus or a prophage, specifically because it's got all of the genes. If I blow that up here, it's got the whole capsid and polymerase and lysing enzyme. That's all in there, and it wouldn't take much to pop this puppy out of there and get the viral genes made and start making viral particles again. So this is called a prophage or a provirus, and the whole cell with a prophase or a provirus in it is called a lysogen. A lysogen. There we go. All right, so I want to talk about the lytic phase right now. Lytic phase. Ow. Hang on. Having a drink of water. Actually, let's not use PowerPoint for this. Let's go back here. All right, let's look at the lytic phase or the lytic cycle for viruses. All right, I was doing E. coli in green. Let's do that. Okay, so step 1 is called attachment. Okay, here's this E. coli cell minding its own business, and along comes this T4 bacteriophage that attaches to the cell. Okay, for this attachment for T4 to land and stay stuck and attached to E. coli, there has to be a receptor on the cell wall of E. coli, right? So there has to be like this little protein here. Let me get this one out of the way here. There has to be this little protein here that will allow attachment of the bacteriophage. we go. So that's actually what makes it host specific. So it's a receptor on E. coli has to bind to the spike protein. And that is exactly what happens with coronavirus as well. Coronavirus has spike proteins that bind to receptors on human cells, especially human lung cells. Human lung cells, heart cells, and I forget what the other one is. One particular mucosa. I'm not sure. I'd have to go look that up. All right, so that's step 1, attachment. Step 2 is entry. Entry and uncoating. All that means is that, okay, E. coli back here, I said that it has to have a gene that lets it get, a gene that makes a protein that lets it get in and out of the cell, in and out of cell. In the case of T4, there is a protein called lysozyme. Okay, well, it's similar to the lysozyme that's in human tears. It produces lysozyme that puts a nick in E. coli's cell membrane. So it's going to do that. So it puts a little hole in E. coli, and now the viral DNA can get in. Okay, now I'm not going to focus on prophage stuff right here, although that can happen, but we're talking the lytic cycle. All right, so we have attachment. Did not mean to do that. Attachment, entry, and uncoating. Uncoating means that the capsid stays out here. Empty capsid, only the viral, in this case DNA, gets inside. Okay, so step 3, we're out of space here. Step 3. Transcription, let's say expression of viral genes. So transcription and translation of viral genes. So these genes get turned into proteins. So they start making viral proteins, including some of the capsid proteins. So they're being made. So 4, now if these are being made, you got to make a copy of the DNA, right? So that would be replication. Replication of the viral genome. So it's going to make more copies of its DNA, as well as all the proteins that it's made up of. What if I put the cellular parts in orange so we can see a difference between the viral proteins and the viral DNA? Something like that. All right. So there are going to be, FYI here, early and late made proteins. For example, some of the early proteins are going to be like the polymerase that needs to be made before you can copy the viral genome. Usually the last thing that is made is this lysozyme, because if the lysozyme is made too soon, it's going to burst the cell from the inside out. So a late protein would be the lysozyme. All right, so let's see. We expressed the viral genes. We made copies of the viral genome. Now they just have to be assembled. So let's say assembly, where all the viral particles get packaged up, put together. Viral DNA gets to be put inside there. Oh, something, by the way, usually chops up the host cell's genome, so that finally when this lysozyme, don't do that. Finally, when this lysozyme is made, step 6, is release, where the cell is lysed. I can't spell. The cell is lysed or bursts. The cell bursts, and the virions are released into the environment. Ta-da! That is the lytic cycle. And next time we will pick up where we left off, and we'll talk about, what do we want to talk about next? Oh, we will talk about different types of viral infections next, acute and persistent, and we'll talk about classifying viruses, how we do it, and we'll start talking about coronavirus in that context as well. So I hope you're having a great day, and I will see you in Zoom. Bye-bye.